Hello there. Today I'm here to discuss first BSc first semester cell biology and genetics paper, syllabus of both theory and practical, affiliated to Bangalore City University of BSc Biotechnology to NEP batch. Okay, now let us go through the syllabus and our university. Right now I'm dealing the syllabus related to the Bangalore City University syllabus for BSc Biotechnology UG. Okay. So this particular syllabus started from the academic year 2021 and 2022. Now let us go through BSc Biotechnology Basic or Honors first semester syllabus. Content of the course 1 Theory DSc T1 BTC 101 Cell Biology and Genetics. Okay, total 56 hours. Unit 1 Cell and Cellular Organelles. Here you need to study about historical perspectives, discovery of cell, the cell theory, ultrastructure of eukaryotic cell, both plant and animal cells, structural organization and functions of cell wall and plasma membrane. Then structure and functions of cell organelles. Here in this, you need to study about cytosol, endoplasmic reticulum, Golgi complex, mitochondria, chloroplast, ribosomes, lysosomes, peroxisomes, nucleus, nucleolus, vacuole, and cytoskeletal structure. In this, you need to study about microtubules, microfilaments, and intermediate filaments. I request you all to write this syllabus content in your first page notebook so that you can keep all this content in your mind. Next, Unit 2, Chromosomes and Cell Division. Here, general introduction, discovery, morphology, and structural organization of centromere, secondary construction, telomere, chromonomer, euchromatin, and heterochromatin. Further, you need to study about chemical composition and karyotype. Okay, in this, single standard and multi-standard hypothesis, folded fiber and nucleosome models. Then further, you need to study about special types of chromosomes like salivary gland chromosome and Lamprecht chromosomes. Here, cell cycle, phases of cell division, mitosis, meiosis, cell cycle checkpoints, enzymes involved in regulation, cell signaling, cell communication, significance of cell cycle, achromatic apparatus, synoptimal complex, senescence, and programmed cell death. In short, we can say this programmed cell death as BCD. So this is all about Unit 1 and Unit 2. Now let us go through the Unit 3, heading Inheritance and Gene Interaction. Here, history of genetics, Mendelian theory, laws of inheritance, dominance, segregation, incomplete dominance, codominance with example, law of independent assortment, test cross, back cross, and non-Mendelian inheritance. Further, you need to study about maternal inheritance, plastid inheritance in mirabilis, Jalapa, then Kappa particles in Paramecium, Petite characters in yeast, sex-linked inheritance, chromosome theory of inheritance. Okay. Then gene interaction, supplementary factors, comb pattern in fowls, complementary genes, example, flower color in sweet peas, multiple factors, skin color in human beings, epistasis, plumage color in poultry, multiple allelism, blood groups in human beings. So all these I need to study under the Unit 3, Inheritance and Gene Interaction. Unit 4, here, linkage and mutation. Here, general introduction, coupling and repulsion hypothesis, linkage in maize and Drosophila, mechanism of crossing over and its importance, chromosome mapping, this linkage map in maize. Then mutations, like types of mutations, spontaneous and induced mutagens, then physical and chemical. Okay, here types of mutations, spontaneous and induced mutations you are going to study. Then further mutagens like physical and chemical mutagens you are studying. Then mutation at the molecular level, mutations in plants, animals, and microbes, and its merits and demerits you are studying. Structural and chemical chromosomal, structural and numerical chromosomal aberrations. Then sex discrimination in plants and animals, concept of allosomes and autosomes, XX, XY, XX, XO, ZW, ZZ, ZO, ZZ types. Then further last content, allosomal. Here you are studying Klinefelter syndrome and Turner syndrome. These two syndromes which comes under allosomal disorder. Autosomal disorder, you are studying Down syndrome and Craig-Cat syndrome conditions. So these are the 4 units you are studying in your syllabus. Unit 1 heading cell and cellular organelles. Unit 2 heading chromosomes and cell division. Unit 3 inheritance and gene interaction. Unit 4 linkage and mutation. So these are the 4 units you are studying in your syllabus of first semester biotechnology. Now let us go to the formative assessment like pedagogy. Pedagogy in the sense the way of teaching or delivering the lecture or information that may be through lecture concept, through presentations, or by showing videos or assignments and weekly formative assessment test. Okay, so assessment pattern, assessment, field report, or a project. This carries 15 marks. Okay, weightage in marks 15. Then test 20 marks. Participation in the class, that is attendance, carries 5 marks. So total you have 40 marks in your theory paper as IA marks. Okay. Then cell biology and genetic laboratory content here for your cell biology and genetics biotechnology paper 1. Code, this is course code DSC P1 PTC 101. Okay. So here your assessment carries 25 marks. Okay. Practical IA marks carries 25 marks and your theory paper carries 40 marks. It means 35 marks you are writing. So you are writing 25 marks of practical and 25 marks you are writing. You are submitting the assessment marks, 25 marks. Okay, so total 40 marks is IA marks from theory. And you are writing your theory paper for 60 marks and IA marks 40. So total 100. Then practical examination you are writing for 25 marks and 25 marks is your internal assessment marks. Okay, so this is all about your internal assessment marks. I hope I made myself clear to you. Theory paper you're writing for 60 marks, 2.5 hours. Okay, so 60 marks from theory paper and 40 marks will be from our side as internal assessment. And in practical, you are performing all these experiments, and in the examination hall, you're performing the experiments for 25 marks, and 25 marks will be from our side as internal assessment. So in this first semester cell biology genetics, these are the experiments you need to perform in your practical classes. First one, operation and working principle of simple and compound microscope, use of micrometry, measurement of onion epidermal cells and yeast, study of mitosis in onion root tips, study of meiosis in grasshopper testes, or maybe onion or maybe Rio flower birds. Then fifth experiment, mounting of polytin chromosomes. The sixth experiment, buccal smear, bore bodies observation, karyotype analysis, human and onion, both normal and abnormal. Abnormal in the human, that you can keep in your mind, the Klinefelter syndrome, Turner syndrome, Craig-Cat syndrome. Okay, that you can keep in your mind. Then eighth experiment, isolation and staining of mitochondria or chloroplast. Ninth one, enumeration of RBC using hemocytometer. 10th one, simple genetic problems based on the theory, gene interaction. Then 11th one, preparation of submission of 5 permanent slides of mitosis and meiosis by each student. So these are the different experiments you are performing in your first semester biotechnology practical paper of cell biology and genetics. Okay, so you can write all these experiments a list in your practical observation book. Okay. Then how we are dividing this particular practical 25 marks assessment. Okay, formative assessment occasion like assignment or monograph, it carries 10 marks. Then test carries 10 marks, and participation in the practical class carries 5 marks. So total 25 marks. You're writing the examination of practical for 25 marks. You're writing. Okay, then 25 marks from our side are as internal assessment. Then what are the books you can refer for the cell biology genetics? Means here some of the books are given here. You can go through the books. Okay, title, for example, Cell Biology of this particular author. Author is very important here. You can focus on this author, and here again, Gene's second, we can focus on Benjamin Levin. Different editions will be there. You can focus on that one, but basic, whatever the notes we give, you can refer that also. If you want, you can purchase all these books. I need 2 or 1 you can purchase. Otherwise, whatever the notes we are giving, no, if you study that one, study material, that will be more than enough. Okay. So if you want to purchase, you can purchase this CB Power Cell Biology Himalaya publication. That is very easy, and easily you can understand the concept, and even you can purchase here the book Genetics. It is PK Gupta. So these are some of the reference books. You can take screenshot and you can study. So this is all about your cell biology and genetics theory and practical paper, along with the reference books list. Okay, so you can visit library also to understand the concepts and to prepare your own notes. If you can't prepare your notes, even I'll give into the class that particular notes also we can prepare. Okay, I hope I made myself clear to you. You be the gift to the world, and thank you for watching this particular video. Thank you, everyone.